So everybody, welcome to today's webinar, coming to you semi-live from Finland. So hi, I'm Dr. Krista Rantanen, and with me today is Dr. Petra Miikkulainen, and we are going to present today. So I'm going to start, and I'm going to do the bits that tells you why. It's the biology bit of today's talk. And when I'm done, so Petra here is going to take over and is going to tell you how to do it. So that's how it's going to run today. As usual, everybody has participated in this numerous times. So if you have questions, please use the chat box and we'll go over the questions towards the end of the presentation. Also, please keep your mics muted, so it'll just be more comfortable for all of us. So okay, we're just gonna go. I'm gonna go off video as well, so you can focus on following the slides, and I do hope you enjoy. So as you know, today's topic is, I'm just gonna put that one away, sorry. Today's topic is anything but oxygen. So we're going to talk about what kind of considerations are needed when growing anaerobic and microaerophilic bacteria. And like I said, I'm Dr. Krista Rantanen, and I'm the Director of Scientific Applications here at the Baker Company. I'm also a visiting scientist at the Francis Crick Institute in London and the lead of Hypox EU, so Community for Oxygen Biology. So this presentation today focuses on anaerobic and microaerophilic bacteria, and it's because these are the types of microorganisms that require special growth conditions when they're studied outside the human body. And I do have a few slides here on anaerobic bacteria, and then a list of or examples of different species, although of course appreciating that many people in the audience, this topic is very familiar, but bear with me. So as you know, anaerobic bacteria can be classified by their tolerance of oxygen. Okay, so we have facultative bacteria, facultative anaerobes, and now this group doesn't really mind the oxygen level, and they can grow either aerobically in the presence of oxygen or anaerobically in the absence of oxygen. So it's a very, very versatile group of microbes that can use oxygen in their metabolism or utilize a reduction of other compounds as a source of energy. That could be like glucose or nitrate or sulfate or something else. So that's facultative anaerobes. Then we have the group of microaerophilic bacteria. So now this group requires a low oxygen concentration, and low meaning somewhere in the ballpark of 2 to maybe 10% of oxygen, but they also often require high carbon dioxide concentration, which would be around 10% for them to grow. And many of these bacteria are those that can cause disease in humans, including Campylobacter and Legionella. Then we have the group of obligate anaerobes, and now this group only grows in anaerobic conditions, and so oxygen is very, very harmful for them. So and many species from this group are part of something called the human microbiome, and I'm going to come back to the concept of microbiome in a moment. So listing facultative anaerobes, as you know, they are, for instance, Staphylococci, E. coli, Salmonella, Listeria. We have the microaerophilic group, which could be Campylobacter, Helicobacter, Lactobacillus, and then those obligate anaerobes, Bacteroides, Prevotella, Clostridia, and so forth. Now, this group in particular, they have essential roles in human well-being and in normal functions. However, in stressed situations where, for example, the body's mucosal barrier is broken, these are the exact anaerobics that can cause severe infections or disease. So from clinical perspective, each hospital with clinical microbiology lab, they need to culture these anaerobic bacteria for correct diagnosis and treatment of infections and what have you. And on research side, then there has been a great interest to study the interactions of these obligate anaerobes and human cells. And it has actually revealed quite interesting mechanisms already where the microbiome affects the pathology of a disease or outcome of a treatment. So this is something that we're going to focus a little bit more on. So whilst this talk is about anaerobes and microaerophilic bacteria, I'm going to touch quite a lot on the cells as well, because whilst we study these and test for these, it's always in the context usually of a human or an animal, and we try to understand how they work and what would be the possible treatments. And so therefore we have to understand the interaction that they interact with human cells and so forth. So microbiome, so as a concept, going back a little bit, microbiome is the lining outside and inside of human or animal body. And it consists of different microorganisms that are listed here, such as bacteria and fungi and viruses and archaea. But in this presentation, for various reasons, I'm going to focus mainly on bacteria. Now, microbiome research, when we talk about microbiome research, it mainly focuses on the behavior, the interaction, and the function of microbiota in human or animal physiology and well-being. And that research kind of tries to understand how the microbiome might affect some pathologies or conditions. Also, very interesting and well studied is the digestive tract or gut microbiome, as referred here, and I'm going to go into this a bit deeper. So the gut microbiome is the biggest community of those obligate anaerobes, and they have various very essential functions in the gut. Gut microbiome is also possibly the most well-studied part of the human microbiome. Also, oral microbiome and urogenital microbiome consists mainly on anaerobic species, although these are much less studied compared to the gut. However, having said that, it is very clear that these two play a very significant role in human health and disease. So let's take a bit deeper look on the gut microbiome, as I promised. So there are literally billions of bacteria and other microorganisms lining the gastrointestinal tract, and possibly up to 1,000 different bacteria species in the gut, so quite a lot. And specifically, when we look at that area, it's the large intestine that harbors the largest ecosystem with anaerobic bacteria around that gastrointestinal tract. Now, these bacteria, they are very specialized and have essential roles in the various functions of human body, mainly in immunity. They also protect against pathogens. They have a very important role in metabolism, as well as in cell proliferation and epithelial barrier function. So if there is research going on and on gut microbiome, it is very important to understand that these kinds of studies need to be conducted in a very controlled environment. And this is something that we're going to talk more in the later part of the talk. Now, gut epithelium, so the microbiome of the gut and the gut epithelium, it is very, very low in oxygen. It's a low oxygen environment. And here we have, well, a very schematic representation of the gut and the lumen. Now, this is the area of the body where the oxygen gradient is the steepest, okay? And I'm just going to walk through why there is such a low oxygen in this region. So as you can see, there is a very steep oxygen gradient here on the right-hand side. And it comes from this very, the oxygen comes from the well-vascularized subepithelial mucosa. And then the gradient goes to the lumen that is practically anoxic. So you can see the well-oxygenated blood coming in, and then the oxygen gradient forming. And that steep oxygen gradient is the thing that creates a very unique physiological demand of those microbes that are in the gut. And the key factors that contribute why this region is so low in oxygen. So the blood flow and vascularization, that's one. So like I said, the subepithelial mucosa that is located just beneath the epithelial layer, it is very highly vascularized as it receives oxygen from the capillaries that are nearby. But as we move deeper into the lumen, the blood flow and oxygen diffusion decreases, and of course that results in lower oxygen availability. And then we look at this border here. So the metabolic activity is an interesting one. So the intestinal epithelial cells, they are metabolically very active because they have roles in things like nutrient absorption and secretion and barrier function. And this high metabolic activity consumes oxygen, which again contributes to decreased local oxygen levels. And then the said microbes. And there is a diversity. The community is very diverse, the microbial community, and they actually compete for oxygen. So three things that contribute to low oxygen: blood flow and vascularization, metabolic activity, and the diversity of the microbe community all create this oxygen gradient. So very, very low in oxygen in the lumen. Now, importantly then, when we understand that there is very little oxygen for these microbes in the gut epithelium, and they need to be there in that environment, we have to understand maybe a bit of a molecular basis for the whole thing. And here we go and talk about HIF signaling for a moment. So like I said, even though we talk about bacteria, we have to understand that it's in a clinical situation. The bacteria in the gut, for instance, they have to interact with human cells. And so therefore, it's important to understand how human cells react to oxygen. And going back to very basic signaling cascade here, introducing hypoxia inducible factor. This is a transcription factor. And so in a normal situation, in an incubator, for instance, in 21% of oxygen, which we breathe in, the cascade goes so that you have a transcription factor called HIF. And if there's oxygen, this group of enzymes, which are deoxygenases, so they need oxygen to function, they do something called prolyl hydroxylation. So they hydroxylate the prolyls in HIF, okay? And this whole group then is seen or recognized by von Hippel-Lindau protein here. And this whole lump then is a signal for the proteasome to be transported to the proteasome and be degraded. So this cascade, remember, happens in 21% of oxygen. So in that incubator. And it takes only less than 5 minutes. So HIF is always produced by cells, yet it's constantly being degraded, okay? However, if we move to conditions that are also in the gut epithelium, which is the low oxygen, could be 1 to 13%. 13% would be in the lungs, maybe not in the gut, so it's a bit lower. But in any case, in that scene, that environment where these bacteria that you want to study, where they reside, there is something called hypoxia or physoxia, so physiological oxygen conditions. And like I said, so the enzymes, the prolyl hydroxylase enzymes, them being deoxygenases, they can't function if they don't have oxygen. So they can't earmark HIF here. And so HIF doesn't get degraded. Instead, it can now move to the nucleus and find a binding partner. It has a binding partner, ARNT or HIF1 beta, whichever you prefer. And together, this dimer binds to hypoxia responsive elements of a gene and starts the transcription. And this is the regulation. So, taken together in high oxygen, which is the ambient oxygen that we breathe, HIF doesn't exist. Say in the lumen of the gut where the microbes work, HIF does get expressed, okay? And it makes a huge difference. Why? Because HIF controls its target genes in those conditions. And as you can maybe see, it's a bit small, but you can maybe see HIF in the center governs or directs or activates or inhibits basically every cellular event, so be it migration or metabolism for energy or other products or growth or apoptosis. So HIF is in the center. So it really governs the cells which these microbes are working together with. And so it is fair to show it like this. So if you don't have HIF, you don't have the control of many cellular functions that should be happening. So this is to remind everybody that HIF signaling is very important, because in this next slide, we're going to talk about HIF signaling. And so it's important that you remember how that works. And now taking back, let's go back to the gut and the lumen and the lining. So HIF gets active, like I just said, HIF gets activated only in low oxygen conditions, okay? And this activated HIF signaling, what it does, it promotes the tight junctions and barrier integrity, because it induces several genes and also production of anti-inflammatory cytokines, as you can see. So if we have HIF, which we do in the up here, where the oxygen is low, and this is the bit from here, we have HIF activation, and it secures, it keeps the barrier safe, okay? So this barrier here is intact, and the lumen of the gut is protected and also separated from the outside. So the microbes inside actually stay inside. So in the normal barrier function, the epithelial layer, of course, it acts as a barrier between the gut lumen and the underlying tissues. And it is this low oxygen condition at the luminal side here that helps maintain this barrier. And one of the ways it does that is it prevents oxidative damage. In a different situation, if we don't have intact HIF signaling, which unfortunately very often is the case when we work in a lab in an in vitro situation. Now the problem comes is that we don't have HIF signaling, we have activated NF-kappa beta signaling. And the problem with this is that NF-kappa beta signaling drives pro-inflammatory pathways here. And what happens then is that it leads to barrier breakage. So you can see there's a gap in the barrier. and it loses the, so in the previous picture, you could see there's the mucus layer with HIF signaling, but with NF-kappa beta signaling, we don't have that layer anymore. And this exposes to the epithelia, for instance, to pathogens. So, and that's a problem, of course, an inflammatory signal. So this is what happens if we study the activity of microbes on cells in conditions that are not mimicking the oxygen atmosphere of the gut. So it highlights the importance of in vitro experimental conditions, because it's not only the bacteria component that is affected by oxygen levels, the human cell component is heavily affected by it too. So that's the problem. Okay, so in summary, too much oxygen, we have HIF dysregulation in the gut epithelial. We're talking about that one now, and in the microbiome. And what happens is that there will be intestinal barrier function abnormalities. So as stated, HIF does play a very crucial role in maintaining the integrity of the intestinal barrier, because it regulates the production of tight junction proteins and also mucins and barrier stabilizing molecules. And so dysregulation of this HIF signaling weakens this barrier. And what happens then physiologically, it leads to increased intestinal permeability, and it's often referred to as leaky gut. And also the immune response is compromised. So HIF does modulate the activity of various immune cells in the gut. So when HIFs are dysregulated, it can lead to imbalance. And then that imbalance leads to imbalanced immune response, and it contributes to conditions like chronic inflammation and, for instance, conditions like IBD. So that's a big problem. Also, the microbial composition is altered if we have HIF dysregulation, which comes from too much oxygen. So the gut microbiota, it can influence and be influenced by HIF activity. So dysregulation of HIF, it can really alter the gut environment, and it might favor the growth of pathogenic bacteria over the beneficial ones that you actually need. So this imbalance, which is called dysbiosis, can make gut inflammation and other gastrointestinal issues far worse. And also metabolic interactions are changed if HIF pathways is not active, because HIFs are very much involved in metabolic processes within the guts, and dysregulation of this can disrupt the metabolic metabolism of short chain fatty acids and other metabolites, which are crucial for the gut health and immune function. So these kinds of disruptions can cause a cycle of compromised gut barrier and then therefore alter the entire immune response. So now we have talked about anaerobes and obligate anaerobes. We have talked about microbiome, and we have put that in the guts, and we have established that there is a key component in the gut keeping this whole thing steady and flowing, and that one component is oxygen. So I want to take a step back now and put this whole thing into context, and here a slide of background. So when we do research, biomedical research, the goal, and I believe we can all agree, is to understand biology so that we can make things better. We want to cure disease, and only way that we can do this is that we correctly mimic the conditions and the components of those conditions. So we can't just take the microbes, or we can't just take the cells of for instance, the gut micro environment to understand what goes on in there. We have to understand the interactions properly, and the surroundings where this works and is greatly affected by And something that is very often overlooked is oxygen, okay? So the thing is, we have oxygen is everywhere, of course, and it's good. It would be hard without, but there's a problem, and the problem is these two dots. These two dots is the problem that oxygen creates. These two dots are the two electrons on the outer shell of oxygen. And oxygen, like any other, would want to fulfill the octet rule. It would really, really like to have 8 of these dots on the outer shell, but it doesn't. It only has 6. So the problem is that therefore oxygen in this uncoupled electron state in most compounds anyways makes it highly reactive. And it's, I mean, we all know this. I mean, everybody's seen rusting. That's the activity of oxygen. Or it can be fast. We've all seen burning. We know combustion engine. There are explosions, and it's all because of the reaction of oxygen. Okay, and it comes from this electron structure, the unfilled electron space in the shell that makes oxygen so reactive. But what we often don't think is that oxygen is similarly disruptive for biomolecules. The biomolecules are not protected. So if you give a biomolecule 21% of oxygen, it is a massive injection of fuel to that biomolecule that it wants to possibly deal with, because a lot of these activities are enzymatic. They just can't, and a lot of the biomolecules are destroyed by using too much oxygen. So taking that together, when we breathe, the oxygen goes in, surprise, surprise, and it's, say, 21%. But then it faces barriers. There are cell walls and tissues and what have you, and oxygen doesn't really diffuse that far. So what happens, and what's the case in vivo in a human being or an animal or whatever, cells and biomolecules are used to having much less oxygen than 21%. And if they have 21%, they cannot handle it. So if we grow cells in ambient oxygen levels, we create unphysiologically high oxygenation and highly variable alterations in oxygen. So now this goes back to our topic today, for instance, the study of microbiome in the gut, how important it is to actually appreciate that the conditions where we conduct these experiments have to consider oxygen. And in many cases, it has to be also anoxic. So let's go back to the picture that I showed you before. So microbiome research has really focused on new treatment options, of course, and developing them further in certain condition or disease, or for new diagnostic techniques. And microbiome has been now understood to be increasingly attractive for many therapies. And we now know for a few years now that gut microbiome health or disease or dysfunction affects various diseases. They can be neurodegenerative, cancer, metabolic syndrome, and various others, as you may have heard. And also a lot of studies, of course, are done on health supporting probiotics and prebiotics and symbiotics and pharmabiotics. But I have a couple of examples also when it comes to therapeutics, when we talk about antimicrobials. And I'm taking this back to relate to the oxygen that we just spoke about, because when we and this goes to the pharma people in the audience, maybe kind of a request to understand how sensitive the oxygen level really is to the outcome of these studies and and potential therapies. And the first one here is antimicrobials. Now, I just read this statistics, and did it really say that in the past 10 years or so, we as a research community, we haven't been able to produce maybe 4 new antimicrobials, therapeutic ones, and was it like 1 maybe that was more potent than the old ones? And of course, this relates to the fact that we are facing a disaster possibly in the next few decades when we don't have working antimicrobials anymore, and then a lot of bacteria gain strength, and we don't have anything to fight them with. So maybe the call for action here for researchers and pharma is to think why the standard susceptibility tests that we perform on antimicrobials still are done in vitro in normal room oxygen. So in that 21%, which I just explained, would be very harmful. And they try to predict in vivo effectiveness of antimicrobial therapy by doing this under room oxygen conditions, because not only of the detrimental effect of too much oxygen on biomolecules, oxygen limitation does decrease the sensitivity of the pathogens to most of the antibiotics. So oxygen limitation, okay, so if there is little oxygen, the sensitivity of pathogens is lower. And if you study these, the sensitivity in high oxygen, you're going to say, well, they're very effective. And then this is made into a drug and it will never work because the sensitivity is lower. So the urge here is to really say that the testing of new treatments has to be involving considering oxygen limited environments, and we'll come back to how that is done a bit later in this presentation. And something a bit more novel, which has certainly moved forward in the last decade, is fecal microbiota transplantation. And we can see here in this slide, there's gut dysbiosis is mentioned. I mentioned it before, if you remember. So in diseases where that are related to the gut microbiome being disturbed and not functioning is also cancer and neuropsychiatric disorders, so Alzheimer's and schizophrenia and so forth. So they have all been shown to relate to gut dysbiosis. Okay, so if then we want to fix this, and the science has come up with fecal microbiota transplantation, FMT. Okay, and it aims to fix the situation and has been shown to function quite quick or quite effectively. And how it goes is that you have donor screening, you have stool collection, packing, and administration. But the problem here is, despite that we know that the absolute majority of gut microbiota are anaerobes, as we established earlier in this presentation, it is amazing that stool processing for this transplantation that is thought to be very effective is now commonly performed in the presence of atmospheric oxygen. It's not to say that it doesn't work, but what happens and what is known is that atmospheric oxygen exposure during this processing diminishes and alters the microbial community. Okay, so then the only understanding from this and action that needs to be taken is that processes like FMT procedure, they really have to be done in low oxygen workflow. So it doesn't just go that you do one bit of the process in low oxygen. It really needs to be a workflow where this is considered. Now, I have said many things here, and I talked about the different types of bacteria and microbes and their need for oxygen or the lack of oxygen. And we've established that in, for instance, gut microbiome, which harbors a lot of these bacteria, is very important in human health, and increasingly we understand that. And so that's all clear, and we understand that oxygen and the lack of is very important for being successful in these procedures. What we haven't heard yet is how to practically work with anaerobes and how we practically can ensure that the workflow isn't only anoxic, okay? It's actually precisely controlled to be so and monitored. And here is the point where I hand over to my colleague, Dr. Petra Miikkulainen, and she's going to tell you how to practically work with anaerobes. Petra, your turn.
Yes, I do see that there is some a slight problem with the slide, but let's not mind that. So thank you, Krista.
That's yeah, not a big problem. You go.
So in these next few slides, I will introduce you to the Baker solutions for anaerobic and microaerophilic bacteria culture. And then after this part, you will have a possibility to ask some questions. So yeah, so a few words about the Baker company. So Baker is an expert in biological safety and precision cell culture and bacteria culture workstations. And the Baker brand is actually made of 3 previously independent laboratory manufacturers, including Ruskin Technologies in UK, and some of you in the audience might be familiar with the former Ruskin anaerobic or hypoxia workstations, or Baker Ruskin brand for the past 10 years. But today we are called the Baker Company, and there are 2 sites of operation in Europe and 1 in North America where the Baker company is originating. So the anaerobic and microaerophilic workstations are still manufactured in UK, and they make a large proportion of our grow portfolio. And this is the lower part of the picture. You see a workstation there. There should also be some text, but the text is missing right now. But in addition to the workstations, like pictured here, we also manufacture high quality biological safety cabinets and clean benches and isolators and various special enclosures and customized biosafety solutions, and these belong into another product portfolio called Safeguard. So together, the Baker company serves customers in different laboratory environments and all over the world. So moving on to the actual workstations and how to precisely control your culture conditions, whether this is a bacteria culture or possibly co-cultures with both bacteria and human cells. So if the workstation method or workstations as a concept is new to you, so few introductory words here. Here in the picture you see a closed system. So it is designed to have a precision controlled environment inside. This environment is both temperature controlled and it is gas controlled for the anaerobic or low oxygen environment. And the work is performed through these round blue glove ports that are sitting on the front screen. Now you don't see any gloves or sleeves attached to the workstation, but of course, when you are working with the workstation, there is some kind of barrier between the open air and then the controlled environment inside. Yes, so like mentioned, the workstation basically combines an incubator and a biological safety cabinet, as we have the temperature controlled internal chamber, and this is designed both for any working steps, handling of your culture materials or samples, but also the incubation. So the materials are not traveling in and out from the workstation, but they actually stay inside the workstation. And yeah, talking about transferring the materials. So everything that is taken into the workstation travels through the interlock on the side. So Krista is nicely pointing out to the interlock space. So this ensures that no excess amount of oxygen, no excess amount of the normal air is introduced into the chamber, thanks to the interlock purge and in between the outside world and the inside of the chamber. Also, we have a single plate entry system on the front screen. So this is a quick and easy method to insert some small materials very quickly into the system. But yeah, the idea, of course, is that transferring the materials or working with your hands in, that does not disturb the controlled environment that we have inside. Yes, so of course, for example, in a research lab, it might be very important to conduct the whole bacterial culture workflow and sample preparation inside these strictly anaerobic conditions. And what is the main benefit for the workstation is that you can handle your plates, you can access the samples, monitor your bacteria growth at any point during the incubation, whereas some other methods does not allow you to check or to play around with the individual samples. Yes, so moving on to the next slide. So very often the anaerobic bacteria are stored and cultured and incubated in these so-called anaerobic jars. So these are these small containers where you have your anaerobic or microaerophilic conditions inside, and then these small containers are placed in a separate incubator for controlling the temperature. The condition inside these jars is usually achieved by a commercial gas packs, as you can also see here in the picture, or a gas replacement method. So the anaerobiosis, or the creation of the anaerobic conditions, it's usually based on a chemical reaction. So bringing in a certain gases that will as a result of a chemical reaction will eat away the oxygen from the environment and usually results in humidity. So the gas packs or the gas replacement method, it usually includes a hydrogen source, and the hydrogen and oxygen are then forming H2O in a simple reaction. So these jars, they might be handy to use if there is only a few samples, but of course there are some drawbacks with these ones, and first and foremost, there is no constant control of the anaerobic or the low oxygen state inside an individual jar. So you will insert your gas pack, and as long as the container stays closed, you can believe that the conditions are as you're expecting them to be. So yeah, another downside is of course the fact that once your samples are inside these containers, you cannot see what's happening in an individual sample plate. And then, of course, any sample handling steps must take place on an open bench. Next slide, please. Inside the workstation, both the work and the incubation will take inside the same place in an automatically controlled anaerobic or low oxygen conditions. And the gas control here in the workstation, yes, we need to create the low oxygen or anaerobic atmosphere, but this is done fully automatically by the workstation, so it does not require a manual work from the user. So only the samples are transferred inside the workstation, and then they are in the optimal conditions. And yeah, depending on the workstation model, you have much more space inside the workstation compared to the anaerobic jars. And there is the possibility also to place some small instrumentation inside your controlled environment. And then you don't need to take the samples in and out. So moving on to the next slide. So I already mentioned a few benefits in working inside a workstation, especially if there is a large number of samples. If there are various different sample vessels, dishes, plates, shake flasks, tubes, etc., or if the research requires some small equipment or some small analytical instrument used in the under the anaerobic or microaerophilic conditions. And yeah, when the samples are all the time kept in the optimal temperature and the optimal growth environment, there is no lag time in the bacteria growth. Yes, so next I would like to move on to the actual workstation models. So very briefly introducing first our small workstation range. So in the next slide, we see our a newly redesigned version of our popular Bugbox workstation range. Krista, can you go to the next slide, please? So, the Bugbox range has been there for 30-something years, and now recently this has got a new look and a new technological uplift. And of course still has the same robust anaerobic or microaerophilic technology behind. So this is currently the smallest workstation with a side interlock available in the market. So this is really designed to be the ideal jump from using the anaerobic jars to start using a workstation. Yeah, without taking too much bench space and being a budget-friendly option for smaller sample volumes. So it has a very compact footprint, but it can fit a lot of samples. If we measure the capacity in Petri dishes, it can fit over 250 plates, the maximum being 270. Just to give you an idea. So yes, absolutely the same idea. You have your glove ports. All the materials are transferred through the interlock on the side, and the workstation is operated from the touchscreen. On the next slide, we have the Bugbox MX. So this is exactly a replica of the anaerobic AX, but this model is also capable for the microaerophilic conditions. And it is easy to switch from anaerobic to microaerophilic mode. So this has the best flexibility for research labs. When you want to work occasionally with microaerophilic bacteria, occasionally with anaerobic, or if you want to do co-cultures with human cells and bacteria. So here, both the oxygen and the CO2 levels can be controlled independently, and the user can set any desired set points between the range shown here on the slide. Yes, and on the next slide, there are a few illustrative pictures on actually using the BugBox range workstation. So on the upper left corner, there is a Petri dish holder taken through the interlock. So this is to illustrate the size of the interlock opening. So it is large enough for different types of vessels or plate formats, and it also allows to transfer a bottle up to a 1 liter through the interlock. Yes, and on the right-hand side up, there is a picture of a single plate entry system and inserting an individual sample plate or some other small materials quickly and gas economically into the chamber. And on the lower side, you can see that the hands are in the workstation. The glove port doors will hang on the sides of the workstation, so the whole bench space is available for working, and the materials can then be incubated and left on the shelf, or if there is a rack system inside the workstation. So yeah, like despite the compact size of the workstation, these models have still a good space to work inside, to do any steps involved in your bacteria culture workflow, sample collection, etc. But I also want to introduce our slightly larger range, so the concept range. If the BugBox model can fit around 250 Petri dishes inside, so this model can hold up to 500 plates. So basically it has double the capacity in the internal chamber. But of course, sometimes or oftentimes you don't have this many plates, but you want to put maybe a small shaker or a cell counter or a small centrifuge or other small equipment inside. So then the concept workstation is a good fit for you. Here we have some advanced features. For example, the removable front screen for, again, placing some instruments inside that are too big to go through the interlock. But yeah, we also have, for example, internal HEPA filtration to keep the internal environment very clean. We have a few different models. So this is our Concept 400. Then in the next slide, we have exactly the same, but with a slightly bigger interlock. And then finally, the third model is our dual chamber, where you have 2 independently controlled chambers. So this can be, for example, different temperatures on each side, or you can even have one side anaerobic and one side a low oxygen or an oxygen control chamber. For example, on one side you can culture your bacteria, and one side have your human cells and do the co-culturing there. Or of course, just simply more space for strictly anaerobic work. Yes, so coming to an end, the concept range also comes, or the single chamber concept comes as a microaerophilic model, and this has been quite popular among the microbiome researchers because of the flexibility of the oxygen and the CO2 control. So if you are interested in placing some even larger instruments inside anaerobic or low oxygen environment, for example, microscopes with oculars, or a plate reader, or even a cell sorter instrument, we also have larger solutions, larger anaerobic workstations, and you can contact us for more information on that. So yes, coming to the final bit. So we have collected here a few examples of the microbiome research performed with our instruments. So only with the Baker or the former Ruskin workstations. So there are over 2,000 publications done with our workstations, and these are just examples from last year or so, and you can see from the topics, and you can see the authors and the author affiliations, that our workstations are used in a wide range of different applications worldwide within the microbiome research. Yes, that's all from me. Handing
Wonderful.
back
Thank
over
you.
to Krista.
Yes, thank you. Thank you. I think I'm just going to stop sharing so that we can actually, show our faces here. And I could actually turn on the video. That might help. Thank you very much, Dr. Petra. So this wraps up our presentation for today. So whether you are an anaerobic or microaerophilic, these are the topics and products that you have to think about and then utilize. So I'm checking. We don't have any questions at the moment, which is okay. So if you should have any further questions, feel free to contact us. So you can find contact information at bakerco.com. So visit the website. Anybody, if you have questions like here, if you want to ask a question out loud, that's also okay. Always appreciate a good conversation. Seems very clear to everybody.
Yes, no raising hands.
Yes, no. Well, that's wonderful. We have been very clear, Petra and I, today. So I suppose then that just leaves us with wishing you a very wonderful day wherever you are, and hope to see you somewhere in the future. So thank you for participating, and see you later. Bye.
Yes, thank you, everybody.